Welcome back, everyone. Happy Tuesday. Hopefully you had a nice, relaxing, restful weekend. Looking ahead a little bit, we're going to finish up reproduction this week today, and on Thursday will be the end of it, and then get into 1 week of heredity next week. So we'll do that in 2 days next week. On Friday, I'm going to have to cancel my extra help because they're allowing us back into the building for an hour and a half or so to just clear stuff out. So I'm going to be doing extra help on Thursday at noon this week instead of Friday at noon. So I'll remind you again later and during the week, but just I'll figure I'll tell you that outright right now. All right, to get into this now, how many daughter cells are created by mitosis? If we remember from last week, mitosis is asexual reproduction. It says so right down here. And that's where you get identical daughter cells produced. You start with one cell, and that is just going to split into two identical cells that are identical to each other and identical to the cell they started with. So there's two daughter cells produced by mitosis. That should be a 2. It doesn't really look too much like a 2, so I'm going to write over. There we go. 2 daughter cells by mitosis. And as for the steps in creating new daughter cells by mitosis, remember the letters P, M, A, T stand for the 4 phases of mitosis, and they each tell you what it's going to be. So the P is prophase, P for prophase, P for packed. The chromosomes condense, the DNA condenses. You finally actually see it as this structure called the chromosome. The M is metaphase. I'm not even going to write phase, I'll just write meta. M for meta, M for middle. The chromosomes line up in the middle of the cell. A is anaphase. A for ana, A for away. The chromosomes start to pull to opposite ends of the cell. And T is for telophase. T for tele, T for 2. You get 2 new nuclei forming. The 2 groups of DNA that are on either side of the cell start to get a new nucleus forming around each one. And then that also will pinch in the middle of the cell and pull apart to make your 2 identical daughter cells that we see up top. So PMAT, prophase, prophase, metaphase, anaphase, telophase, for your 4 stages of mitosis. So we saw last week that some reproduction takes place asexually with one parent only, and that's one parent cell now splitting to make identical copies of itself. We know that that's not the only option because that's not how we work, right? We use mitosis to make body cells. We can use it to repair damage, to heal cuts or lost cells, to just kind of turn over old cells. We want to get rid of them. We replace blood cells pretty frequently and commonly, or just to grow, right? We can get bigger by just making more and more of our own cells, identical copies, and that actually makes us larger. But humans also reproduce sexually. So on a larger scale, to actually make a new person, we do use sexual reproduction with 2 parents forming offspring who have a combination of the parents' DNA. So it's combining genetic material from both parents as opposed to just one cell at a time splitting and making identical copies of itself. And the process that does this is going to be called meiosis. So mitosis is asexual, meiosis is for sexual reproduction. This is going to make the reproductive cells. I know it's kind of tricky. It's one of those cases of, why didn't they just think of 2 words that are more different sounding? Unfortunately, we don't make those rules. But meiosis is now where you're, again, creating the sex cells needed for sexual reproduction. So these specialized cells, that is going to be the sperm in males, and the egg, or the ova is another word, in females. These are called gametes. So we saw last week that body cells, the one that are not reproductive cells, which is pretty much everything, are called somatic cells. That's a fancy term for them. And these are the opposite ones. So the reproductives, the eggs and the sperm, are known as gametes. That's the word that just overall means the reproductive cells in different animals or plants. And these cells are going to have half as many chromosomes as any given normal typical body cell. So if a body cell normally has 50 chromosomes, a gamete would have 25. A body cell would have 10 chromosomes, a gamete would have 5 chromosomes. We'll see what the correct number for us as humans is in a little while. But question now is, why would these egg or sperm cells only have half of the correct number of chromosomes in a cell? Why would that make sense? Why would it be okay for them to only have half of what you'd expect any other cell to have? To get the answer to that question, we need to talk more about the difference between different cell types. So a cell that has the full 2 copies of every chromosome, one from mom, one from dad, is called a diploid cell. We saw that in people, human beings, we have 46 chromosomes, 23 from mom, 23 from dad. This would be a diploid cell, di meaning 2. 2 copies of every single type of chromosome. That means that diploid cells are, again, the body cells or the somatic cells, if we want to get technical with that. And we sometimes can describe these as what we call 2N cells, which N just means the number of copies of chromosomes. So you don't really hear this a lot, but that's one way you might see it, described in some way. Sometimes that 2N cells are, again, your body cells with 2 copies of every chromosome. And these are the cells that are going to be made by mitosis. So these are normal body cells. They can do all the normal life processes that cells need to do in order to maintain homeostasis. And again, they're going to be made when you need to replace a cell because it's old or damaged or anything. It's just identical copies, asexual reproduction. One cell will copy itself, copy itself, copy itself, going from 1 to 2 to 4 to 8 and so on. So these are your diploid cells, the normal kind of cells. And you see again at the picture there at the bottom, 2 sets of every type of chromosome. The alternative to this type is haploid cells. And these are only going to have 1 copy of each chromosome. So this is going to be a little bit different, half as much as normal. And these are going to be the cells, again, with only half as many chromosomes as you'd expect, and these are the sex cells or the gametes. Now, if the diploid cells are referred to as 2N, we refer to the haploid cells as 1N. They only have 1 copy of every chromosome. So instead of the normal body cells where you have 1 copy from mom, 1 copy from dad from every type of chromosome, in these cells, the sperm or egg cells, you'd only have 1 copy. You either have a dad's chromosome or a mom's chromosome, depending if you are an egg cell or a sperm cell. Where are these cells coming from? Are you coming from mom? Are you coming from dad? That's going to determine whose chromosomes you're actually getting. What's the origin of these chromosomes, where they're actually coming from. So again, 1N cells, these are made by the process of meiosis. And since these cells don't have the normal amount of DNA, they only have half, they can't maintain homeostasis on their own. So these cells cannot survive on their own. An egg cell or a sperm cell cannot ever grow into a full person. They can't just survive without doing something else. And that thing they need to do is actually to combine with each other. So an egg cell and a sperm cell are 2 different haploid cells. Each one only has half the correct number of chromosomes, half the correct amount of DNA, and they will need to combine with each other. When those 2 cells merge and fuse together, they each bring their half the correct amount, and that'll let them make 1 normal complete cell now that has the exact correct amount of DNA that you should normally have. That full cell will then make more copies of itself by mitosis a couple of million times during the course of any given lifetime or more, to keep making more and making more and making more and growing and growing and growing to become a full entire person. And here we have Dr. Hank McCoy explaining it to us. Not mutant signs, L signs. So I crossed out the word mutant there. I really don't like this artist. And yeah, that's all I'm going to show you, because you shouldn't need to look at this artwork anymore. As we start to talk about meiosis, just again, remind yourselves that mitosis starts with 1 cell. DNA is copied, so you have twice as much DNA as you need. And then that 1 cell splits apart so that each of the 2 new daughter cells gets the correct amount of DNA. You copy everything first, make twice as much as you need. So when you break apart, everybody gets the correct amount. That being said, I want you to think of this new process of meiosis as just 2 rounds of mitosis. That's really all it is. It's like you're doing mitosis once, and then you're going to do the whole thing again. In mitosis, DNA is copied to make twice normal amount. The cell divides to give each of the daughter cells the correct number of chromosomes. In meiosis now, the DNA is going to be copied, the cell will divide, but then each of those cells will divide again. So this is going to make 4 daughter cells. And each one of them is only going to wind up with half of the correct number of chromosomes of a typical average body cell. You copy it so everybody has twice as much, cut in half once so you get to the normal number, and then when you divide again, cut everything in half again, now you only are going to have half the right number. And that's how you're going to make the reproductive cells. It's a fine process because mitosis make identical body cells, but meiosis, the whole point of it is to make reproductive cells, the egg and the sperm, that only need half the correct amount, because they will then go on to combine with each other and make a full normal cell again. This first part we're going to call meiosis 1. So this is going to be the first round of divisions. And then we'll look at the second part after that. Meiosis 1, the second part will be called meiosis 2. Each part has the same steps as mitosis, the same prophase, metaphase, anaphase, telophase. Everything's going to go pretty much the same way with one little difference. But again, we're going to do it twice, so 2 rounds of this whole thing. Before anything can happen, the cell needs to make a copy of its DNA just as before. So the DNA is going to condense into the chromosomes, as we've already seen in mitosis. In this cell here, we start with the normal 46 chromosomes, and we'll see 3 types of chromosomes. So here we've got 2 reds, 2 blues, and 2 greens. And again, notice there are 2 of each type. These are the copies that you get from mom and dad. So this will be now 1 copy of red from mom, 1 copy of red from dad, 1 copy of green from mom, 1 copy of green from dad, and 1 copy of blue from mom, 1 copy of blue from dad. Now the DNA is going to be copied, and that leaves us with 92 chromosomes in total. So you had 46. I only actually show you 3 here because I'm not going to draw 46 different copies. When you start with 46 chromosomes, you then copy it to get to 92. That's twice the amount of normal DNA. That's 2 copies of each of the normal 46 chromosomes. So in the second picture now, we see that each chromosome has been copied, and they go and kind of hang out together. They attach together and make that X shape with matched up sister chromatids, we call them. So now we have a double copy of dad's red chromosome, a double copy of mom's red chromosome, a double copy of dad's green and mom's green, a double copy of dad's blue and mom's blue. They're not labeled as such, but that's just what's going on there. So we have a double copied pair of each chromosome from both parents. Good so far? This is how mitosis works. So far, it's all exactly the same. This is exactly what we saw last time. We didn't go into as much detail with this in the picture I showed you for mitosis, but this was kind of setting up mitosis. This is all sort of getting into prophase. This is the one place that's going to be different now. In mitosis, the X's, the copied pairs of sister chromatids, are going to line up single file in the middle of the cell. So for mitosis, metaphase would look like this. You'd have all these X's right down the middle of the cell. Notice in meiosis, it's a little bit different. In meiosis, they're instead going to line up in two lines. Both double copies of the red chromosome line up next to each other. So instead of going one single file right here, now mom and dad's double copies line up next to each other here for red, and then both of the blue copies from mom and dad line up, and then both of the greens, and so on for all the different chromosomes in your body. So meiosis, the first part here, you're going to line up double file. X's line up side by side in these pairs. That means now that when the spindle fibers pull them apart, when you've got these little things over here, and they're attached to these chromosomes, they're pulling this way, and one's going to pull this way. In mitosis, it would have split the X. So this X right here would have become a line here and a line there. In meiosis, it's going to be a little bit different. Now the X's are going to separate as intact whole X's. Each X pair of chromosomes will stay in one piece, and it's only this whole intact structure that's going to move apart and separate from each other. So let me get my black ink turned on here. But this X would move in this direction as one piece. This X moves in that direction as one piece. The blue moves here as one piece. Blue moves here as one piece. They don't actually separate into their parts. They stay as one whole little X shape. When the cell splits now, each daughter cell may not look exactly down here like it did when we started. So this cell right here doesn't look exactly like it did up top, but all these same amounts of DNA are there. So up top we had two of these little red things. Right here we still have two reds. They just happen to be paired up in an X. We had two greens. We still have two greens. They just happen to be paired in an X. The amount of DNA is correct, even if it doesn't look exactly like what it started out as. But that's fine, because we're not done yet. We only went through one part. We only went through one phase. This is the first part of meiosis. Again, we call it meiosis 1, the first round of the process. In prophase, we see the pairs, P and P. In metaphase, they line up in the middle, M and M. Anaphase, away, A and A. In telophase, you now form two new cells, T and T. Now we're going to jump right back into it and do the whole thing again, starting right from this exact spot for meiosis 2. We're starting right where we left off here with these two cells that we just made from the first round of meiosis. The nucleus doesn't even reform in each cell. It just steamrolls right ahead into the next part, which we now are going to call meiosis 2. So these intact pairs of sister chromatids, the X's, from the first round of meiosis, immediately go and line up again in the middle of each cell. Now the rest of the process is back to being exactly like mitosis. This is going to be metaphase. They're in the middle. So it's almost right in. And really don't even have a whole prophase. Goes right into the next round. From here, the X's themselves are pulled apart, just like we saw in anaphase before, with the sister chromatid pairing separating into two chromosomes again. So at this point, take a look at these cells and compare them to where we started. Once again, change my ink color. At the beginning, we had two reds, two blues, and two greens all the way up in this top picture. One of each color from mom, one of each color from dad. Now, after two rounds of divisions, we're left with only one of each color. So I see down at the very bottom here, one red, one blue, and one green. By my math, that's half the normal amount of DNA. In humans, that would mean 23 chromosomes per cell, whereas a normal body cell should have 46 chromosomes at any given time. The cell made two copies of everything. Dividing once gave each the two daughter cells here in the middle the correct amount. So again, it looks a little bit different than you'd expect, but it's the correct amount of DNA number-wise. But we divide a second time, and that gave each of these final four daughter cells that we ended up with at the end of the process, end of the day, half of the correct amount. And that's what we want, because one of these reproductive gametes, one of these cells at the bottom that we just made, can at some point combine with another one, either an egg or sperm, depending on what this one is, to reestablish a full set of DNA and become a brand new person during sexual reproduction. By the nature of genes combining from two parents, sexual reproduction creates variation in offspring. While mitosis and asexual reproduction create identical copies, Offspring from sexual reproduction are different from their parents because they're combinations of their parents' genes. But meiosis actually makes things even more mixed up and helps make offspring as different as possible. One way that meiosis creates variation, difference from parents, is called crossing over. This is something that happens in the first metaphase of meiosis 1, when the X's, the copied sister chromatids from mom and dad, line up in pairs in the middle of the cell. Remember, there are two files, they're on a double file lining up. It's not all a single file. When the chromatids are next to each other, when the X's are next to each other, they twist around each other or cross over and trade pieces. So mom's chromosome and dad's chromosome, the X's of each one, actually sort of wrap around each other right here. The ones that are right next to each other actually wrap around each other and will trade little pieces off. And just notice in the picture how they trade equivalent pieces. Not like this piece goes to this chromosome and then a different piece goes somewhere else. It's a one-for-one trade. In this case, it's the bottom parts of these two chromatids, the X's, that have switched places. This is a random process that swaps out pieces of one chromatid for another. So when it's done, the chromosomes eventually separate. We see that one chromosome on the right here is all of dad's genes. It's all blue. We see one that's mostly dad with some of mom's genes down at the bottom. So we have one here that's mostly blue, but it's got a little bit of this purplish color. We see one that's mostly mom's genes, mostly purple, with a little bit of dad's blue color at the bottom. So those genes. And we have now one that's entirely mom's genes. It's entirely in purple. So this mixing and matching of genetic material is just an extra source of differences in a sex cell, which can then be passed on to the offspring. Instead of just having only mom's genes or only dad's genes, you can get, again, entirely dad, entirely mom, mostly dad with a little bit of mom, or mostly mom with a little bit of dad. And this is in each person. So really, in terms of how the kid is going to be affected, it's actually a little bit of grandpa, or entirely grandpa with all of grandma, or some of grandpa and some of grandma, or mostly grandma and some of grandpa. In the getting case, it's just lots and lots of different combinations of genes. So it's not just this or that. It's a lot of different things. This is only showing one little place where they can cross over. You could have some mixture here, and then some here, and some here. Infinite number of possibilities of how these things are actually going to be trading gene pieces. So again, these are 4 potential options of the chromosome that can then be part of a sperm or egg cell. But realize this is only one type of chromosome. There are 45 other chromosomes doing the exact same thing, each one making 4 possible versions after crossing over. And the way in which genes are traded is totally random. So this creates an astronomically large number of possibilities of gene combinations. And each one of those can be then passed on in any given sperm or egg cell. And considering your body is making millions of these cells all the time, the possibilities of what gets passed on in terms of information to the children is just really endless. But this again leads to an entire scrambling of these genes and just huge numbers of possibilities mathematically. Crossing over adds a lot of potential variation to our genes and is one reason for why we're all so different from anyone else who's ever existed. But the bigger reason is this, what we call independent assortment. This one sounds a little weird, but we need to think bigger picture here. Have you ever heard someone say something like, she's got her mother's eyes or something like that? Where do we think those mom's eyes actually came from? Where did the genes come from? They came not so much from the mother herself, but from the mother's mom or the mother's dad. It's things that get passed on throughout the generations. When chromosomes line up double file during metaphase, when crossing over is going to happen, if it's going to happen, they don't line up in any set arrangement. There's no such thing as all the chromosomes I got from my dad sent over here and all the chromosomes that I got from my mom sent over there. The chromosomes just go wherever they happen to go. Once again, completely randomly. So in this picture, we see 2 possibilities. On the left, the chromosomes from a person's dad are on the left and their mom are on the right. So right here we see all the blues on one side and all the reds line up next to each other, double file on the other side. In the other picture, dads are on the left and mom are on the right for the bigger chromosomes. So blue on the left, red on the right there. But it's reversed for the smaller chromosomes. The red are on the left and the blue are on the right. Either one is just as likely to happen as the other of these 2 options here. It's a total coin flip how this works, but keep in mind, once again, this is only showing 2 different chromosomes. These are only chromosomes 1 and 2. What happens to the other chromosomes, pairs 3 through 23? Do the math. How many different combinations of left or right are there if things are just lining up on any given side all the way down for 46 chromosomes, 23 pairs? Anyone actually do the math and figure out possible combinations here? It's heads or tails, 23 different times. How many combinations are you going to get for that? I didn't do the math either, but I looked it up, and the claim is that it's 8,324,608 possible combinations of chromosomes, and that's without anything actually crossing over, trading pieces, realize this is only in one parent. The same thing's happening in the other parent too. So that's another way to add more math in. Add 4 possible combinations of chromosomes after crossing over is taking place, with totally random ways that they may trade genes during crossing over. Then it's going on in both parents' bodies. You have all these numbers to begin with. I'm not even going to try to figure out how that math would work. The bottom line is this: every one of us is a mathematical impossibility, unlike anyone who has ever or will ever live, and that's a really, really cool thought. And I know I covered up that last bit of notes there, so I'm going to backtrack and get rid of that if I can. It's not letting me. I don't know why. I'll show you in the next slide. I'll repeat it again. I guess it didn't like that I had written over something in the slide. So here's that last little bit I kind of jumped past. Same thing I was saying. It's just tons and tons and tons of possible combinations. Just bigger numbers you can ever possibly imagine. So that's it for new stuff today. Let's do a couple of quick comparisons here, mitosis versus meiosis, and then I've given you a handful of practice questions, and we're done for today. We've got mitosis on the left, meiosis on the right, and let's compare. Number of divisions. Mitosis is going to be asexual. I'm actually going to write that right up here. Oh, I don't want that color. It's supposed to be green. Let's go back and change that. Mitosis is going to be an asexual process. Be helpful. I can actually write. There we go. Asexual. And meiosis is going to be making cells for sexual reproduction. That's better. Okay. So the number of divisions in mitosis to make 2 identical daughter cells is going to be 1 division, making 2 daughter cells. We have 1 cell here, and it's just going to split off, make 2 identical copies. So right there again, identical copies. What kind of cells are these going to be? Well, this is when you're just making more of the same, more of the same, more of the same. So this is when you're going to make identical body cells. Remember, mito does mitosis. Mito, like a toe on your foot, does mitosis. Types of cells, these are going to be diploid. They have 2 copies, 2x, 2n was really the abbreviation, but 2 times the copies of each chromosome. So those are diploid cells, di meaning 2. And in humans, we do this to repair tissue or to grow or anything like that. For meiosis on the other side, sexual reproduction, that's going to now be just basically 2 rounds of mitosis. It's going to be 2 divisions. So you go from 1 cell, making 2, and each of those cells gets turned into 2. So you got 4 cells overall. Making 4 cells created. There's going to be different cells. That's where you do the 2 rounds of it. The fact they line up the X's in pairs first means you're not getting just identical information each time. They're getting mixed up. You also, again, now we know have crossing over independent assortments. So you're getting lots and lots and lots of variation, which is a great thing for us. And types of body cells, this makes the gametes. That's the sex cells. That's the sperm and the egg. We'll see a little bit more about that next time when we finish up our chapter with sexual reproduction. And types of cells, these are now going to be haploid cells because you get half from a sperm, half from an egg, and that's going to equal a whole for an entire new cell. That's going to make now an entire new person. So this is going to be sexual reproduction. It's used for in living things. Looks like I gave you 4 practice questions. Let's do them right now. A cell that has 2 copies of every chromosome, we could refer to as 2n, is called what? What does it mean when you have 2 copies? Well, the prefix for 2 is di. That's going to be diploid cell, and that's most of our body cells. Those are the somatic cells. Remember, when we're talking about these things, basically if you're not a sperm or an egg cell, you count as a body cell. Doesn't matter what type of cell you are. Every cell in your body should have the right number of chromosomes unless it's a sperm or egg. So where do these copies come from? They just get copied from each other. It's mitosis. And these are going to be identical copies. So 1 cell gets copied if it's a stem cell. If it's a cell that's already there, it doesn't matter. So 1 cell will make a copy of itself to make 2. Those 2 cells will make copies of themselves to make 4 to 8 to 16, 32, and so on and so on and so on through your entire lives. But 2 copies is a diploid cell. Different version of the same question. A cell that now only has 1 copy, we call it 1n cell, is called a what? Diploid cell was 2, so we know it's not going to be that because di means 2. So what's the one that means 1? That's the haploid cell. If you forget this, you can look at these choices. Polyploid means many. Poly is a lot, so that's not going to make any sense. We know haploid is 1. So this is when you have either an egg cell or a sperm cell, and this sperm cell is now going to go into that egg. We'll see in a couple of days. This one is half the right amount of information. This one is half the right amount of information. This is now going to be a full complete set, and that's going to make all the right amount of DNA. Why are these cells important? They help us reproduce. And in the case of animals and plants, reproduce sexually. And this adds a lot of variation. Variation is a very good thing because it makes us all different. It lets us evolve. It lets us change and grow and improve, and also get rid of some stuff we don't really want. That'll be next chapter and a half or so we see. But for now, that's all we got to know. This question says, a sperm cell of an alligator has 16 chromosomes. What is the total number of chromosomes normally present in the stomach of an alligator? Stomach cell of an alligator. Now, just remember, sperm cells and egg cells always have half. So our gametes always have half the correct amount, and body cells or somatic cells always have a full amount of chromosomes. So if we're talking about a sperm cell, an alligator has 16 chromosomes, well, that means then that 16 is half of whatever the number should be in a body cell. So we're looking for a stomach cell in an alligator. That counts as a body cell. Again, if you're not a sperm or an egg, you count as a body cell. So if 16 is the normal number for a gamete, that's half of what we'd expect in any body cell. So that means a body cell would have to have 16 times 2, which equals 32 chromosomes in a body cell, and that's our answer right there. If we know how many chromosomes we normally have in a somatic cell or body cell, we can cut it in half to find our sperm or egg number, and if we know how many we have in a sperm or egg, we can just double that to figure out what should be in a body cell. So based on what that last question was, what do you really think I was going to give you here? Same exact question, but opposite, kind of reverse in this case. So now we have it going the other way. Remember, gametes, sperm or egg, are half the right number, and body cells, the somatic cells, are equal to 1 of the whole number, the entirety of the whole number. So if each body cell now of a chimpanzee contains 48 chromosomes, we know a body cell should have 48 chromosomes. How many chromosomes would be present in a gamete, a sperm or an egg cell made by that chimpanzee? We're just going to cut this number in half, and that's going to give us 24 chromosomes in whatever that gamete happens to be. Just doesn't matter which one. All we care about is that it's half the normal body cell number, and that's going to be 48 divided by 2 is 24. We are done for now. Go do the forms for today, and then you got some worksheets posted in OneNote. That's going to be due next Monday, 5/18, along with the stuff for later in the week on Thursday. Quick scheduling thing, again, as I mentioned earlier, no extra help this Friday at noon because I can't do it. I got to get back in the building. So I'm going to do Thursday instead. That's you don't even know what day that is. What's this calendar say? Is it the 14th? That is the 14th. So it'll be 14th at noon on Thursday. And I'm just expecting that going forward. I'm going to change my schedule things. Now that I have office hours on Monday, I don't think there's really a point in doing extra help on Monday. Also, that may be a lot for me to do in one shot and for anybody else who would be coming to anything. So I may have to change that day. And if I do that, I'll probably end up changing the days that I give you work. So I got to figure that out, but I'll let you know whatever happens going forward. In any case, get this done now, and I'll see you in a few days.